Okay, we're going to be talking about cells, chapter 7. It's kind of a long chapter here, so bear with us. We'll get through it all. First, we're going to talk about the cell theory and what that is, because we're going to be talking about cells in general, so we'll start there. The cell theory says three things. Number one, it says that all cells come from other cells, pre-existing cells. It says that the cell is the basic unit of structure of all living things. And it also says that all organisms are composed of one or more cells. There are two main types of cells, prokaryotes and eukaryotes. Prokaryotes are cells that do not have a nucleus and they have no membrane-bound organelles. And we're going to talk more in detail about those in a little while. An example of that is bacteria. Eukaryote cells are like you. They are cells that have organelles, membrane-bound, and they have a nucleus. Plants, animal, fungi, protists, all of those are examples of eukaryotes. Two big major types of cells, and they are divided up based on whether they have a nucleus or not. When we're talking about cells, we have to know how to use a microscope. So we need to look at the different types of microscopes and know how to use them. First of all, you need to know that when you take a microscope, you need to hold it by holding its neck at the base and then bringing it up to your chest to keep it so that it won't fall anywhere. The first type of microscope is the type that we have in our classroom. It's called a light microscope. And what it does is it bends light through an object so that we can see the tissues in it. It can either be a lamp, a light, or it can be a mirror that reflects the light from the room up there. We can see living cells and tissues in it. That's the benefit of it. They're very inexpensive and they're mobile. They're easy to move around. But they're kind of limited in that it can only magnify things 1,000 to 2,000 times. And that's a big magnification. I believe the ones in our classroom can only magnify a maximum of 450 times. The eyepiece at the top of the microscope has a power of 10. And then each one of the objectives, low, medium, and high, that are on the nose piece or the wheel that goes around, has a power to it. You take the power of the objective times the power of the eyepiece of 10, and that will give you the magnification of the microscope. Here's an example of some of the things that we can see, and these kind of look like the cells that we will have looked at if you're doing this at the end of the chapter, or we will look at. Next is a scanning electron microscope. It uses electrons. It bounces them off the surface of the object, and it scans them. It gives us a nice 3D image of the object. It's really nice because you can have a magnification of 10 to 20 times power. But the problem is that you cannot look at live specimens. That when you bombard it with the electrons, it kills them. They're also very large and very expensive. But look at these images here. They give you beautiful 3D images of the cells or the organism. Next is a transmission electron microscope, TEM. Trans was one of our prefixes, and it means through. And so this takes electrons or across or through. Electrons travel through the sections of the specimen, which is really cool because we can magnify them 100 to 200 times power. This is where we can actually see a lot of those organelles that we can't see with our microscopes. Again, the problem is that the organisms die, so you can't see them alive, interacting, responding to stimuli, that type of thing. Here is an example of what you can see. You can see the nucleus is in there, and the different types of organelles. One of the main type of organelles that we're going to talk about is the plasma membrane, and this is what surrounds your cell. It is inside the cell wall when we're talking about animal cells, and it is the outer region of the cell when we are talking about animal cells. They're very flexible, and it protects the cell from its environment. Two other ways to describe it, or the names for them, is cell membrane and phospholipid bilayer. The function of the plasma membrane is to allow nutrients into the cell things that it needs to undergo its daily functions, such as water, glucose, oxygen. And it allows waste and products of cell metabolism to leave, so like the waste. So it brings in what it needs and lets out what it doesn't need, or the waste. The thing about the membrane is that it is selectively permeable, and that means that it's picky about what it lets in. It's only going to let certain things in. So some things will be allowed to come in, some things won't be allowed to come in. The structure of the membrane is it's made with phospholipids, and we're either done this activity or you will be doing an activity where we have phospholipids, where we have the head and the tail. The phospholipid is made of glycerol, phosphate group, and 2 fatty acids. The head, or the top part, is what we call hydrophilic. It likes water. And the tail is hydrophobic. It does not like water. So again, the head is polar, it's hydrophilic, it's water-loving. The tail is nonpolar, hydrophobic, water-fearing. So the phospholipid molecules are going to arrange in 2 layers with the tails facing each other. And they do this, when they face each other like that, they form a phospholipid bilayer. They have 2 layers of the phospholipids with the tails in between them. So please keep in mind that that whole 3D structure of both of those layers is the cell membrane. It is not like just a single line like we have always drawn it to be when we're drawing a cell. Because of the nonpolar inner layers of the cell, this allows the cell to keep its structure in water and so it doesn't dissolve in water. We will either have done by the time you listen to this, or will be doing an activity where we arrange phospholipids in water in different areas so that you can understand how they are going to arrange themselves, with the head being liking water and the tail not liking water. Other components of the membrane is it's made of cholesterol. This helps stabilize the proteins. It keeps the tails from getting tangled up inside of each other. There's proteins in the middle, and these are needed to move substances or waste materials through the membranes. They attach to the cell membrane, to the intercellular structures, to help identify cells and chemical signals along with surface carbohydrates. This is a great picture of the plasma membrane. Please note that it is very 3D in structure. We've got 2 layers of phospholipids, the heads being the yellow parts here, the tails being the black. These purple areas here are what we call protein channels. So these big proteins, things can attach to them and move through. So the things that are too big to just move through by diffusion will have to attach to the proteins and then move through from a high concentration to a low concentration on that. Again, here is a great example of what it would look like in water. We have the 2 layers, and it wraps around itself. There are 2 large domains of living things, and we talked about those already, prokaryote and eukaryote, and they're divided up based on whether or not the cells have nucleus or does not have a nucleus. So prokaryotes, examples are bacteria and archaea. They are extremely small. They are smaller than their eukaryotes by about 16 times, and they are structurally simple. That's very important for you to know. Prokaryote cell structures, it has a cell wall, and that cell wall is made of lipids, carbohydrates, and proteins. It has no cellulose in it. It has a plasma membrane, which encloses the cell, a mesosome, which is a folded area in the plasma membrane that helps secretion and copying of chromosomes. When things are folded, there's a lot more surface area on them for chemical reactions to occur. It also has a very simple singular chromosome, circular double-stranded DNA. There's just a singular chromosome in them. There's a nucleoid region. This is a general area where chromosomes is located. And there's also plasmids, or smaller circular DNA molecules. This is what a prokaryotic cell looks like. This flagella, or the tail on the end, will help with movement. But if you notice, there's not a lot of internal structures to it. There's no set nucleus, etc. And it looks like a fortress. It has a cell wall on it to protect it. That's why it's so hard to kill bacteria. There's great diversity in prokaryotes. They are used in many, many ways as decomposers. Bacteria as decomposers. They help with the nutrients cycle. They can be photosynthetic, which means they can make their own food. And they can also be a source of antibiotics, which is kind of interesting because the antibiotics are used then to kill the bacteria. Most bacteria are beneficial to us, but there are some that are detrimental to us. The next domain, or the next large category, are eukaryotes. They are everything but bacteria. They are a little bit larger than bacteria, but again, they're small. We need microscopes to see them. What you need to know is that they are internally complex organelles. And an organelle is any part of the eukaryotic cell that has its own structure and its own function, similar to what our organs are like in our body. Why do we have organelles? Well, we have organelles so that many reactions occur and there can be a division of labor. It can be separated from each other. So you need to have these different areas where the jobs can occur. First, we have our cell wall. They are found in plants, fungi, and prokaryotes. They provide support and function, protection, and they are outside of the plasma membrane. This looks what a plant cell looks like. There are two things that a plant cell has that animal cells do not. It has our cell wall here, and it also has these green chloroplasts in them. The green chloroplasts are there to capture the sunlight so that we can convert that sunlight and use it for energy to create food. Also, the cell wall is what gives it its structure, and it protects it. Cytosol is the semi-fluid that is in the cytoplasm. It surrounds and bathes all the organelles. It's mostly made up of water, but there's protein in it also. The cytoplasm is the cytosol plus all the organelles. In the past, when you were younger, you probably learned it to be the liquid of the cell. But it's not technically just the liquid of the cell. It also is the organelles. The nucleus is the genetic control center of the cell. It is enclosed by two membranes, a nuclear envelope, and it contains your chromosomes or your DNA. The nucleolus, which is inside the nucleus, is involved in cell division and RNA production. We also have an organelle called the endoplasmic reticulum, and these are tubes or channels that connect other organelles. It's snuggled up next to the nucleus. There are two types: rough endoplasmic reticulum, covered in ribosomes, and smooth endoplasmic reticulum, where there's no ribosomes. So this is like a channeling system that we can get things where they need to go. Here is a good picture of what one would look like. Ribosomes are small, they're sphericals, and they're found throughout the cell. And this is where protein synthesis occurs. If you notice on the rough ER, there was ribosomes there. Protein synthesis can occur on these ribosomes. Then they can go into these channels of the endoplasmic reticulum and get transported where they need to go. Also, we have an Golgi apparatus or a Golgi body, and this is a system of vesicles or sacs, and they're next to the ER, and what they do is they do the final assembly of the proteins, and they transport, package the materials, and send them throughout the cell. Here's an example of what a Golgi apparatus looks like. Notice that it's flatter and more sac-like looking than the endoplasmic reticulum. Vesicles are spherical membranes that pinch off of the Golgi body. This carries the materials to the other organelles and to the plasma membrane to export things. There are lysosomes in the cells, and these have enzymes that break down the food and the old molecules in old cells, etc., for recycling. It's like the digestive system of the cell. The vacuole contains waters, enzymes, and pigments, so it's there to store the water. There's plant cells have one large vacuole that dominates the cell, and animal cells have several small vacuoles. So here's an example of how large the vacuole is in a plant cell. Chloroplasts are the green that you saw in there, and they are double membrane. They have the location where photosynthesis occurs. They're in plant cells and other autotrophic one-celled organisms. When we start talking about photosynthesis, we will talk about the complexity of these chloroplasts, that they're not just round green things. And this is also a electron microscope of what a chloroplast looks like. Mitochondria is the powerhouse of the cell. It has double membrane, and it's where ATP is another word, is energy, so that you know that. This is where the energy is made and is found in animal and plant cells. Here's an example. It looks what they look like. If you notice, there's a lot of folded areas here. Remember, I told you before, when we have folded areas, that's so that we increase our surface area, so we have more areas that we can have chemical reactions occur. Centrioles are barrel-likes, and these are pairs that are involved in cellular reproduction. They are found in animal cells. Cytoskeleton and other things is the internal skeleton. It helps organize structure and the activities of the cell. This is what it looks like. It looks like a bunch of microtubules throughout the whole cell. That kind of like our bones. It functions to support cell movement, cell signaling. Microfilaments help in movement in cells. Intermediate filaments anchor our organelles where they need to be. Microtubules guide movement of the organelles through the cell and allows for movement of cilia and flagella. These are all composed of protein. Cilia are short extensions that we'll find on the outside of the plasma membrane, and they're made up of these little microtubules, and they will move. They can be used for the cell to actually move, or it can be used to move materials. For example, we have cilia that line our respiratory tract, etc., on all those cells, and those cilia will trap things and move things where they need to be. And this is kind of what they look like under an electron microscope. Flagella are long extensions, like in that prokaryotic cell that we looked at. They're long extensions of the cell surface. They're composed of microtubules, and they help the cell move through its environment. Here's examples of the flagella, the long ends there. This one on the bottom right also has some cilia. What materials do cells need to take in? What materials do cells need to get rid of? How do cells do these things? They do it using a concentration gradient. They move things from high concentration to low concentration. We've talked about this before in terms of diffusion, the movement of particles from a high concentration to a low concentration. For example, when we took our air freshener and we sprayed it at the front of the room and we watched it go through to the back and timed it. It took a while, but it went from that high concentration to the low concentration. We're going to talk about osmosis, and that is the diffusion, same principle, high concentration to low concentration of water across a selectively permeable membrane from high concentration of the water to the low concentration of the water. So here we go. If you look at these orange dots are the salt. So the water is going to go from where there's a lot of water to the not so much water. So the more salt I have, that means the less water I have on the right side. So the water is going to move from the right to left here. Why does this happen? What controls it? Well, water always wants to move in any direction to make it an equal concentration on both sides of the membrane. A concentration gradient is created when we have an unequal distribution of the water or of the particles. So once we get that gradient created, that water is going to want to move, or those particles are going to want to move. Please always remember, though, that water molecules, particles are always moving. They don't just miraculously stop because they've reached equilibrium. What happens to cells in different types of solutions? So we're going to talk about isotonic, hypotonic, and hypertonic solutions. In an isotonic solution, the amount of dissolved materials or stuff is equal inside and outside of the cell. The concentration of water is also equal inside and outside of the cell. So osmosis still occurs, but the cells retain their shape because the amount moving in and the amount moving out is the same amount to keep it equal. Iso means the same. And here's what would happen. We have 10% salt inside, 10% salt out, which means I have 90% water on the outside, 90 on the inside. It's going to keep going in the same direction. Both directions, I mean. Hypotonic solutions, this is when the concentration of stuff is lower outside the cell than inside the cell. Because by having more stuff inside the cell, the amount of water is less inside and greater outside. That means the water is going to move into the cell to balance it. If the water continues to keep gushing inside the cell, it may cause it to lyse or to burst. Remember, lyse was one of our prefixes and suffixes, which means destroy, so that makes sense that that's the word they would use. Plant cells become turgid. They do not lyse. Hypotonic solution looks like this. I have more stuff, 20% salt inside, 10% salt out. So I have 90 water out, 80 water in, which means water is going to move in. Hypertonic means that the concentration of stuff or dissolved materials is higher outside the cell than inside the cell. What that means is I have more water inside my cell because I have less stuff in there. That means water will move outside of the cell, and animal cells will shrivel up, plant cells will wilt. Here's what happens here. 10 salt inside, 20 salt outside, 90, 80. So it's going to go from the 90 to the 80. Same thing happens with these blood cells. Here's a great picture of your cells when your water is leaving your elodia, right? Because your cell membrane is starting to shrink, cell wall's not, and so all of that is coming closer together. Usually these would be spread out to reach your cell wall. So how do these things move? By diffusion, we talked about. And facilitated diffusion is the passive transport of materials over the membrane using transport proteins. They move from areas of high concentration to low concentration. It requires no energy, but it uses the channel proteins or carrier proteins. Active transport is in all material movement that we've talked about already, is always been going down the concentration gradient, high to low. In active transport, we're going up. We're going up the steps, up the mountain, using energy. So we're going from lower to higher concentration, and this requires energy. It's called active transport. So what's happening is we've got these items above that is a lower concentration, and we want to make it go into the cytoplasm. So it hooks up to one of our protein carriers, and then it is pushed through the protein channel and through. And it takes energy to do that. So active transport is the movement of materials against concentration gradient, requires energy, and it uses carrier proteins. If I want to transport large particles, what can happen is I can use my plasma membrane or my cell membrane. And what happens is, first we have endocytosis occur where the cell surrounds and takes in the material from its surroundings. It brings the material inside to the plasma membrane and pushes it through to the other side where it undergoes exocytosis, where it shoots it or excretes it back out from the cell. Both parts of this require energy. This is a great picture of how this happens. In endocytosis, this is your plasma membrane right here. Your plasma membrane takes it in, or cell membrane takes it in, moves it over to the other side, and then spits it back out. Energy, it requires energy at both sections. Here's a great plant cell for you. an animal cell. We already had a couple of plant cells throughout here. Here's our animal cell. All of our organelles, no cell wall, and no cytoplasm. No chloroplast in here. So that wraps up our Chapter 7 notes. There are a lot of them in here, so please listen to this a couple times to help you on your midterm or for your Chapter 7 tests. Good luck.